Phase 1 clinical trial bioanalytical support, speed versus quality. A sponsor receives their first phase 1 PK data on a Friday afternoon. By Monday, their biostatistician had flagged a coefficient of variation above 20% on a critical time point. The method had never been cross-validated for that matrix. The FDA query came 6 weeks later, and that's it. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Laboratories, where we provide bioanalytical support for Phase 1 clinical trials, the work that stands between your compound and the clinic. What is Phase 1 bioanalytical support? Phase 1 bioanalytical support is the scientific infrastructure beneath your clinical decision making. It's the validated method that quantifies your drug and its metabolites in biological matrices, plasma, urine, cerebrospinal fluid, with the precision and traceability regulators require. Before a single subject is dosed, someone has to build and qualify the analytical bridge between what happens in a patient and what appears in a data package. That bridge is bioanalytical support. Most sponsors understand this conceptually, where it gets complicated in the intersection of speed and rigor, and that tension is exactly what Phase 1 exposes. The data generated at this stage will be cited in your IND, referenced during Phase 2 dose selection, and examined in your NDA. Errors made here do not stay in this study, they propagate. Why speed matters in Phase 1? Phase 1 trials operate on compressed timelines for a reason. Every week of delay in a first in-human study carries an opportunity cost, competitive landscape shifts, capital burns, patient access deferred. PK and PD data from Phase 1 inform dose escalation decisions, sometimes within days of sample selection. If your bioanalytical turnaround is slow, your clinical team is flying blind. Fast data drives faster decisions. That is not a marketing claim, it's an operational reality at the dose escalation committee level. When a lab returns validated PK results within 24 to 48 hours of a sample receipt, investigators can adjust cohort dosing with the real evidence rather than protocol of conservatism. The hidden risk of moving too fast. Here is where most sponsors underestimate the risk. Speed without validated methods is not speed. It's a deferred failure. A mid-sized biotech came to us after their early method development had been outsourced to a low-cost provider. The method looked functional on paper. When we ran incurred sample reanalysis, the failure rate exceeded 30%. The study had already enrolled 12 subjects. Every sample from that cohort had to be reanalyzed. The program lost 11 weeks, not because the science was hard, but because the corner was cut during method validation. What a less experienced team often misses is the difference between a method that works in your hands and the one that holds under regulatory scrutiny. The FDA does not grade on effort, they grade on data integrity. The regulators expect, what regulators expect? FDA's 2018 bioanalytical method validation guidance is explicit. Selectivity, accuracy, precision, matrix effects, dilution, integrity, stability under relevant conditions. These are not optional elements. For new molecular entities, the guidance expects validation to anticipate the analytical challenges of your specific molecule. Protein binding instability in biological matrices, endogenous interference. Generic validation packages generate FDA queries. Study-specific, scientifically justified packages do not. What regulators are increasingly focused on is not just whether your validation was done, but whether it was designed for your molecule in your matrix under conditions that actually reflect your clinical study design. That distinction matters when the agency reads your submission. How modern labs deliver both. The analytical technology available today makes the speed versus quality trade-off far less severe than it was a decade ago. High-resolution LC-MS platforms, automated sample preparation workflows, and LIMS integrated data capture allow a well-resourced lab to generate a validated, audit-ready data at a pace clinical timelines require. We have worked with molecules that present real analytical complexity, compounds with endogenous counterparts, highly protein-bound analytes, metabolites that require separate quantitation. Building a validation that holds under those conditions from the start, not retrofitting it after a regulatory query, is what separates program that moves cleanly through phase 1 from those that stall in the middle. Choosing the right CRO partner. 3 things separate a capable bioanalytical CRO from a merely compliant one: scientific depth, regulatory fluency, and timeline accountability. Scientific depth means your partner has seen your class of molecule before and anticipates its behavior. Regulatory fluency means they write validation reports that preempt questions, not invite them. Timeline accountability means committed turnaround windows in writing held to without exception. The cheapest provider rarely delivers the lowest total cost. Factor in the cost of a failed ISR, a delayed data package, and an FDA information request, and the economics looks very different. Real world relations from phase 1 trials. Early planning is the single largest predictor of phase 1 bioanalytical success. Programs that engage their CRO at protocol design before samples are ever collected proceed more cleanly than those that hand over some samples and expect the lab to improvise. A method that was built with your formulation, your dose levels, and your sampling windows in mind performs better under real conditions than one adapted after the fact. Quality first is not slower. It's the shortest path to clean data packages. Key takeaways. Phase 1 is where the analytical foundation of your entire program is established. Speed matters, validated methods matter more, and a partner who builds those methods with regulatory intelligence for your molecule, your matrix, your study design is what actually moves a program forward. If you are planning a phase 1 study and want a direct conversation about bioanalytical strategy, then reach out to Resolve Mass Laboratories. We would be happy to have that conversation with us. Our technical team would be happy to have a discussion with you. Thank you so much for watching this session and see you in the next one. Until then, goodbye for now.